My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. We have a good one for you guys today. I have Dr. Neil Ford, who is a professor emeritus from the biology department at the University of Texas in Tyler. And this is going to be one of those episodes that I've done with a lot of people. These are kind of like, a lot of the pioneers in the world of snake ecology and herpetology. These people who have just had great careers and are, kind of towards the end or later in their career. And we're just going to have a great conversation about the entire career and how Neil got to where he is today. So looking forward to this one. Welcome to the podcast, Neil. How are you doing today?
Doing great.
Good. And I really like to start at the very beginning and kind of figure out how people got into this, what a lot of people in the world might think of as an eccentric interest in things like snakes and herpetology. So I like to figure out where that began. So let's go all the way back to your childhood, and maybe talk to us a little bit about kind of where you grew up and whether this seed or this interest in these animals was planted then, or if it's something that kind of developed a little bit later in life.
I've tried to figure this out for a number of years, and I really don't know. I grew up in Ozarks of Missouri, and in those days you did get out and wander around even when you were young, and I would walk in creeks or whatever in terms of exploring. I don't remember ever finding any snakes early on, but I do remember the very first one I caught, and that we were at a lake, and I was walking down a creek looking for snakes, flipping rocks and things like that. I have no idea how I got to know to do that, but I did, and turned up a worm snake, and they have that beautiful belly, and I took it back to our trailer or whatever we were staying in, and my folks let me keep it there for the couple days we were there, and then I had to let it go, and I remember being very upset that I had to let the snake go, but my folks were pretty good about not inhibiting me too much in terms of my interest in I don't know why it was snakes as opposed to other animals, but it was, and so it just kind of grew from there. We had a shed in the backyard, and I built some cages and caught snakes and put them in there, and it wasn't heated, so I had to let them go, and as the summer was over, and but when I was about 16, they finally let me keep one in the house. I think it was a speckled king. But at any rate, whatever they did, they don't know, because I've asked them, and I don't know. But I'm interested in snakes, and it's kind of driven me the whole way of my career. I'm incredibly lucky. I mean, how many people get to study snakes your whole life and get paid for it too, and get to go all over the place to collect, and so I really can't answer your question as to how I got interested, because I've tried myself.
Yeah, well, it sounds like that spark, kind of caught fire, pretty young. So was that first king snake you had, the one you keep in the house, was that an animal that you caught in the wild, or is that something that you ended up, getting through a breeder or something like that?
Well, they didn't have that. They didn't have breeders in those days. If you were going to keep something, it was something that somebody caught if you didn't catch it. Again, living growing up in the Ozarks, there are plenty of woods around and plenty of opportunities to find snakes. I don't know how I caught that one. I remember catching that one, but I remember catching other ones. I remember I was with my dad exploring a cave, and as we got out and walked back to the car, there was a ribbon snake, and I caught it, and it was real fat. And I thought, oh, maybe it's pregnant, and then all of a sudden it started throwing up a frog. And my dad he was definitely not a fan of snakes. So when it started to open its mouth, it was, oh, you said it wouldn't bite. Well, it wasn't biting; it was throwing up a frog, which it did, and that was obviously kind of an interesting thing to have happen. I don't remember it having a big impact on me, but it was neat to see. And there were lakes around that had water snakes that I could always go catch. You could always find those. So I caught it myself.
Did you see many timber rattlesnakes when you were on your childhood adventures in the Ozarks, or
not there? When we moved to Kansas City, Kansas, and I was at the University of Kansas for my undergraduate, and so on the drive back, there were hillsides that had rocks and things like that. And I guess Joe Collins told me about one spot, but at any rate, I went to a spot that somebody had told me about, and it had a lot of copperheads, and I was looking at those, flipping rocks, looking at those. Well, I turned up about a 4-foot timber, and was going to catch it and take it back because they'd asked me if I see any, bring them back. We're going to put them in the museum on display, live ones. So I said, well, I'll catch this. Well, the only bags I had were about 2 feet long. And this is about a 4-foot long timber. And so it was, and I was by myself out on this hillside. And I managed to get in there finally.
You had a snake hook or tongs or something, I'm assuming.
I must have had a hook or something. Somehow or other I got it in there. And I was on the way home rather than back to the university. So I took it to the house, and just as I was walking in the house, I bumped the bag against the door jamb, and it started rattling. And so my dad heard that, and he knew what it was. He said, "Get that dang thing out of here." So I ended up going back to the university early that weekend.
Yeah, that's great. So you mentioned that you went to Kansas for your undergrad. But let's go back. So you're a child. You have this interest in snakes. You're starting to keep snakes. Did you, as you were kind of going through high school and thinking about moving on to college, did you realize at that point that, this was a potential career track that you could potentially be working with snakes? And did you go to Kansas for that reason?
That was not in my head. You just work with snakes and as a living? No way. I thought maybe I grew up in Missouri and they had a pretty strong conservation program there, and I thought maybe I'd be a park ranger or something like that. I didn't realize that you could study snakes. And what happened, it was really fortuitous. I had a lot of serendipitous things happen, but they assigned me a roommate in my freshman year in the dormitory. And this guy happened to be best friends with John Simmons. And John Simmons was an undergrad who'd gotten hired to preserve frogs and things like that over in the reptile range of the museum there at Kansas. where Bill Dolman was, and all of them, and those. It was a fairly well known facility, and so he introduced me to John, and then John introduced me over time to the other folks in the reptile range, and a lot of them were the grad students, but Dolman was there. I mean, he was in charge, and I had been assigned they just give you a counselor for your career, and I had an English professor as my advisor, and so I asked Dolman if he'd take me on, and he said that he would, and so I got to start hanging around the museum and the grad students and so on, and we were so again, it was just very fortuitous. And then John and a couple others undergrads wanted to take the herpetology class that Bill Dolman was teaching when I think we were like juniors or seniors, and he wouldn't let us do it. He said we wouldn't because it was a grad course too. But he said I'll design a 1-hour class of readings that you guys can come over to my house. We just went to his basement, and he would bring in grad students to talk about their research and give us papers to read, and all of a sudden it was like, so this is what it's all about. You can pick a project and go on to grad school and get your degree and get a job in studying snakes. I still didn't realize it quite then, but that's what was happening, in terms of that. And so again, it was just fortuitous thing that we asked him to let us take his herpetology class, and we didn't get to do that, but we got to do the other, and I knew a lot of these grad students over the years. Then from that point in time, I'd meet them at meetings, and they'd be people that I had learned about at University of Kansas, because it was pretty much a hotbed of herpetology at the time, and there were a fair number of them working with snakes, and so I'd get to. One of the things I did was just offered to go on field trips if anybody was going out, to collect. And I remember going out with somebody to collect water snakes, and he was looking at the band numbers on them. But probably the thing that came out of that was the most important to me was I took a course from Harvey Lillywhite. And Harvey was a physiologist who worked with snakes and animal behavior too. And at any rate, I took a class from him, and sometime during the semester, he said, if anybody's interested in going on a field trip and helping me next summer, just come talk to me. And so I did. And he was going to Southern California, where he did research on the effect of fire on the snake populations there, and I somehow arranged to go along to help. This was in the Laguna Mountains of Southern California, and he didn't have any money for us. We had to pay our own way, and so I didn't have any money, but I had a coin collection. And I sold a coin collection so I could go to Southern California and work with Harvey Lillywhite, looking at the effect of fire on snakes.
Did you ever have a chance to go out to any of the long-term snake research at a University of Kansas? Or?
Well, I didn't there, but later on, I did an NSF grant with Ann Bronikowski, And Anne works in that long-term population of garter snakes that Steve Arnold started studying, where they were looking at the two populations, or two ecotypes, I guess. One was up in the prairies, and the other one was down around the lakes. And I can't remember which one happened first, but I went out and studied, or just helped with those, because garters are my thing. I mean, that's the species I've worked with the most, or the group I've worked with the most. And then Anne came and visited me in Tyler. And the funny thing is, we don't have any garter snakes in Tyler, in Smith County, the area I live in. They don't occur here. So we have ribbon snakes, but no garter snakes. And so she came down, and I had kind of switched to studying cottonmouths, because they're really abundant and easy to work with. And she had some interest in those because they were long-lived species, and she was interested in longevity and how senility and things like this as it relates to reproduction, which is what she was doing with the garter snakes up there in California. So that one, actually, now that you mention another one that started early on, really my first trip was with Bob Henderson of the Milwaukee Public Museum, and I don't know exactly. I guess he met him at the museum at Kansas too. But at any rate, he invited me to go along to Haiti and work with the snakes that they were getting off of islands. There were islands off of Haiti that had never been surveyed because it was hard to get to them. And so he had planned a trip down there, and I was interested in the vine snakes that occurred there, the Euromacer, and I got permits to bring back 3 species of Euromacer and did some behavioral work with them. But it was really my first trip, other than with Harvey. It was my first trip out of Kansas and out of the country, and that was kind of an eye-opening one, actually.
Huh. I bet eye-opening, meaning from the biology perspective, or more like human culture-related things in Haiti, or all the above?
Yeah, more human. An incredibly poor country, and I didn't know basically how to behave. I mean, it was like, how do you deal with folks who are starving to death and this kind of thing? So, yeah, it was eye-opening in that way more than with the snakes. The snakes were great and lots of fun to collect. Although it was difficult to collect, because as soon as the children in the neighborhood figured out what you were doing, they would go collect everything, and you wouldn't have a chance to even go look yourself. And we pay I think we paid $1 for each snake and maybe $0.25 for every lizard, something like that. And they just overran us with animals that, in terms of preserving them, which is what Bob was doing with the animals that were coming in. And then I was collecting the Euromacer from them. But it was, it was an eye-opening experience for somebody who was like 21 or however old I was.
Yeah, it sounds like must have been. So you're doing your undergraduate in Kansas there, and you've had this class that Duhlman helped set up and interacting with these grad students. Sounds like you're starting to get an inkling that, like, oh, I can study snakes for a living. And so where did you end up going from Kansas after you finished your undergrad?
Well, I asked Duhlman where I could go, and he suggested Bill Dagenhard, who was I think in New Mexico, Arizona, and Henry Musinski was at No, it was Roy McDermott. I'm sorry, Roy McDermott was at Tampa, and then looked at. Let's see, it was Carpenter, Chuck Carpenter in Oklahoma. Well, what happened was Degenhardt had too many students and wouldn't take anymore. And this was for grad school. I was asking him where I should go. And then I went and visited Henry, Roy in Florida, and they took me out in the field there. But they didn't have any money to pay me to go to grad school. And I had gone to a meeting down at Oklahoma and had met Charles Carpenter there. And he had a grant, and basically that's where I ended up going and starting on animal behavior as opposed to physiology, which is what I thought I'd be doing. But started working on pheromones, trail pheromones that snakes use to find females, that kind of thing. And that again was just by accident. He happened to have a grant, and the other things fell through. And part of it was I was not a great student, and so I ended up getting in, but only on probation. So I started at the University of Oklahoma on probation. And one of the things I feel kind of proud about is that Chuck years later told one of the other students of our group at the time that he was proud that I'd done so well. Maybe starting a little on the weak side.
Yeah, I had the exact same thing. I started my master's degree on probation because my undergrad was just pretty weak. I think in today's world, it would have been hard for me to get into graduate school or even hard to get into undergrad back in the day. So anyway, so you go to work with Carpenter. Are you working on garter snakes at this point, or what's the species you're working on?
Yes, yeah. Mostly worked on ribbon snakes. They were fairly abundant, and I looked at. Basically, I built an apparatus so I could videotape where females were leaving a trail, a pheromone trail, because you couldn't see it. I mean, it was just chemical in the skin, and they were leaving a trail, and then I would release a male into the apparatus in the arena, and when it encountered where the female had been, I made a little drawing of it with acetate, and I had a video camera to videotape where she touched. At any rate, I could overlay the male's trailing, and it would trail right on top of where the female had been. And so it was basically a bioassay of her production of the pheromones and his interest in trailing too. And so I looked at seasonality of it. I looked at species specificity. I went and got other species of Thamnophis and tested whether they could tell each other's trails apart, because you would expect they would if they're sympatric organisms. If they're together, then they should have different pheromones, right?
Yeah.
And so, sure enough, I got started on that particular program there, looking at species specificity, looking at seasonality, and that's basically what I did for my master.
For example, would a male ribbon snake follow, another, like a species of garter snake, or did you find that they were really able to identify the different species? Hey everybody, I just want to take a quick break and tell you that the Orianne Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.orianne.org to learn more about our work and how you can get involved.
Well, in my PhD work, I found out that I went to different parts of the country and collected garter snakes, and what I found there was some of the closely related species, if they were by themselves in the area, if there weren't any other species, they would get confused if you used some other species from a different region. If it was closely related, like I used Butler eye and I used radix, two of the plains and short-headed garter snakes, and they couldn't tell each other's trails apart, except in the area of Milwaukee, where they both species occurred together. When they occurred together, then they had evolved the ability to tell the trails apart, or they changed the trail. I really didn't know what the chemistry was of it, but they would be able to discriminate the other species if it was in the same geographic area, where because you wouldn't want to waste all your energy following a female and exposing yourself to a predator too. And if she's the wrong species. So
exactly.
If there's any chance of that, then you're not going to follow it. Yeah. One of the interesting things about this whole I did this for a long time. This was my first area of research, even after I came to Tyler. But it was a total serendipitous situation again, because when I was out in California in the Laguna Mountains, and we were looking at the effect of fire in certain areas, the way we were doing that is we were looking at tracks across these sandy roads. The area didn't get much rain, and it was very dry and dusty. And the snakes, when they crossed the road, would leave a track that was easy to identify. And we could actually tell some of the species apart by the way the track looked. And at any rate, one time we ran across a track that was like a double track. It was a track across the road. And then there was another one that was kind of right on top of it. And it would get off a little bit. And you could realize there were two tracks. And one was right on top of the other. So I asked Harvey what was going on there. And he explained to me that there were some papers back, German papers back in the 1920s, looking at trailing behavior in garter snakes in the lab. And he explained that the females were giving off a pheromone, and explained the whole thing to me. And I was like, oh. So when I got to Oklahoma, Chuck asked, one of the first things he asked me, what do you want to work on? And he was one of those professors that didn't tell you what to do. He let you kind of come up with what interested you, and that was really important. Turned out to be very important to me. But when I got there, I didn't really have a clue of what I wanted to work on. I didn't have enough background to really know. But I'd seen that with Harvey in California, and so I said, well, I'd like to study snake trailing behavior. And he said, all right, here's some papers that you can read. And so he dug up a couple things out of his library, and where people had done some preliminary stuff just to show that there were these chemicals being left on the ground that the males would follow. And so again, it was just totally serendipitous that I ended up doing that. But I ended up doing it for my PhD too. And when I came to to Texas since there weren't any garter snakes here. I went to South Texas and worked with another species from down there.
What? Did you leave? Where did you end up going for your PhD? Did you stay in Oklahoma?
Like I know, I could have stayed in Oklahoma, but one of the other PhD students kind of came to me and said, "Look, you didn't do a great job here. You really need to start over," and suggested that he had somebody that he'd worked with when he was doing his master's. This guy was on a postdoc wherever his name was Ron Gratz. And Ron said, "Look, you need to go get a new start. Go work with somebody new." And so we had a meeting, a herpetology meeting down in Tulane, in New Orleans. And so we were, as in New Orleans, what do? You go, out in the evenings. And so I was at some bar somewhere with some of the other students, and Ron introduced me to Doug Taylor, who had worked. He was a professor at Miami of Ohio, and so we're sitting there at 2 a.m. in New Orleans and talking about projects, and I told him about my interest in snake trails and that kind of stuff, and he said, "Well, we've still got one position open in our PhD program. Why don't you apply?" And this was getting pretty late in terms of getting in. But I did. I went back and filled out some paperwork and sent it up, I guess. Or at least Doug kind of probably pushed it on through. But turned out that position was given to somebody else. But they had somebody who was working on his master's, or was going to start on his master's, but he had flunked his biochemistry class and didn't graduate. So they couldn't take him. And again, serendipity here, that position opened up and I got that one. And so I ended up starting my PhD program at Miami of Ohio.
Oh, great. Plenty of garter snakes up there too. So
and there were, yeah.
And so you continued your research on, trailing in snakes. Any other interesting things that came out? You talked a little bit about, kind of the species differences. Anything about like seasonality or anything else you learned, kind of throughout the course of researching this trailing behavior that's worth mentioning?
Well, I think what happened was nobody else was working on this area, and that actually was kind of a good thing too, because anything I learned was kind of new. So yeah, I did some seasonality. I did that actually with ribbon snakes, and what I found there was that there were 2 seasons to the trailing behavior. They breed in the springtime, and so they would show some strong trailing then. But there was a second period in the fall when I would get some trailing too, and I don't know why they don't breed in the fall.
Would it be to, I'm sure you've speculated this, but would it be to potentially find like overwintering sites or?
Yeah, I think I did talk a little bit about that. I think following each other around does if you don't know where to hibernate, you may end up running into one. Yes, that's such an obvious answer. I probably did think of it, but I actually don't remember right at the moment.
Mm-hmm. Yeah.
If I did, but I do remember it being a little confusing. And then at the biggest one was doing this geographic stuff, because that took a lot of work. I went to Colorado to collect them, and you have to catch them while they're still producing the pheromone. So you've only got a few weeks to collect, bring them in the lab, and test them before they start to lose the behavior. And so I used Radix from Colorado. Went there, and Hobart Smith helped me collect collect up there. Got me spots to go. And then, but I went to Illinois, where two of the species occurred together, radix and sertalis. Oh, and then in Ohio, I got butler eye, and it's related to radix. And so basically was able to set up a series of different trials, but I had to do it pretty quickly. So I did manage to go to those places quickly, collect animals. That's the nice thing about garter snakes, is that they tend to be pretty abundant when you find them. So it was possible to catch enough animals to bring in and do the projects. And the nice thing about that was, I ended up having several different kinds of projects that I published on it, and I was invited to go to a chemoreception meeting. So not just a snake project, snake meeting, but I got to go to Oxford in England to give a talk on my research, where with people who were looking at lion dung odors and all sorts of different things, and so met a lot of folks that were related to or who studied chemoreception as a field. And so for the next few years, I got several papers related to that. That was mostly as I came on down to Tyler. I did my PhD pretty quickly. I did it in 3 years.
Oh, wow.
Yeah, and ended up coming down. Well, I took 3 years on my master's, so I was kind of in a hurry. I didn't really want to.
So you went straight from your PhD then down to Tyler.
And that's the only job I've ever had, really.
Yeah, no, that's great. And you continued that line of research that you were talking about, but I know you've worked in other areas, and maybe start, you talked about, where you worked most of your career there in Tyler, that you didn't have garter snakes, but that you did a fair amount of work with cottonmouths. So talk a little bit about, how that developed, kind of your research program around cottonmouths and some of the things you studied there.
Well, I don't remember exactly. It had something to do with the fact that I had all these females in the lab, and a lot of them would have babies. And Carpenter instilled in me, you always take data when you see things. So I measured all these babies that were born over the years, and it was kind of a confusing problem in that there was variation among sizes and numbers that didn't make a lot of sense, or at least I had trouble figuring it out. And so I gave a talk at a meeting where I talked about all these babies that had done some stats looking at size and offspring number and things like that with several species. And in the audience was Rich Siegel. And Rich was interested in the same problem when he was doing his PhD at Kansas with Henry Fitch, and he was interested in some of that too. And he'd gotten a postdoc at the Savannah River Ecology Lab, and he had some funding with it to bring somebody along. And he asked me if I'd like to. Well, during that meeting, we talked a bunch, and we got along real well. And so he said, "Hey, get a species." And actually, I had one at the time. These checkered garters from South Texas, was working with. But at any rate, I had a nice colony. He said, "Well, bring it to the Savannah River Ecology Lab. We'll set up a facility for it, and we'll look at the impact of diet." Because what we thought was going to happen was that the number of offspring or the size of the offspring were correlated to how much food the snakes was getting. So in a year where they were getting lots of food, maybe they'd have more babies, or maybe they'd have bigger babies. Real simple stuff, but nobody ever really tested it experimentally. So we brought the animals there, hibernated them before we went there, and then we put them on different diets and looked at them. We got a paper in ecology out of this.
Wow.
Yeah, which was really neat. And then Rich got a grant from, I don't even remember, but it was a Louisiana state grant, and it gave us money to go out and collect another population of checkered darters besides the ones that were in South Texas that I was collecting. We went out to Arizona, where there was another group of the same species, but so we brought it in, and it behaved differently. The first group, the ones from Texas, you give them more food, they have more babies, and they're the same size. And that's the simple aspect or answer to it. The ones from Arizona, you give them more food, and they have bigger babies, but not at the same number. And so we ended up hypothesizing it had a lot to do with weather and the evolution of these strategies. So at any rate, Rich and I have published a lot of stuff together on the evolution of reproduction and phenotypic plasticity. And that's what we did for the next, I don't know how many years, but we did a number of projects on garter snakes. And the cottonmouths happened by accident. Tyler happens to have lots of bottomland hardwood forests and stuff like that, and I had gotten invited to do the vertebrate surveys of a new wildlife management area north of town, north of Tyler, that was a bottomland hardwood forest, and it had lots of snakes. And so I did a 3-year project just getting them they needed raw data on the mice and the birds and the snakes and everything. So I did that for them. But it got me up there working with the cottonmouths. So I ended up doing kind of the same thing with them, bringing them into the lab and looking at their reproductive strategies. And as a species that only has very few offspring. If you get more food, you're not likely to be able to have another full size neonate. So what we hypothesized was that they would. Well, actually, it had been hypothesized by others first, but the idea that you get more food, you're going to have bigger babies, because you're not going to want to have if you only have enough food for, say, a half-size offspring, you're probably not going to do that. That wouldn't be as evolutionary smart to do. Instead, you would make your other ones a little bit bigger. And I didn't end up testing it on the cottonmouths. I ended up testing it on an Egyptian snake called the diadem snake, and they laid really big eggs. And sure enough, instead of you give them more food, they would have bigger babies instead of more. And now the cottonmouth stuff just happened that they're real easy to work with. They're very abundant. There are a lot of other folks in the U.S. that work on cottonmouths too for the same reason. They're a great model organism. And so I published several field studies, mostly on those, and that kind of got me to know several other folks that I collaborated with, like Ann Bronikowski.
Hey everybody, I just wanted to take a break and tell you guys that snakes are one of the most persecuted groups of animals in the world, but we are dedicated to changing that. The Orianne Society works every day to ensure there's a future for these amazing animals. If you care about snakes, wildlife, or wild places, and you like what you've been hearing on the Snake Talk podcast, support us today by donating on our website at www.orianne.org. Were there other kind of major research themes you had throughout your career at Tyler? You've got kind of the phenotypic plasticity and feeding, and you've got the, pheromone trailing. Were there other major themes, or did you continue primarily on those throughout your career?
Well, one of the things that happened here is it's a very small university, and it was about 2,000 when I came, and it wasn't a research. There was very little interest in anybody doing research.
Is there a graduate program there, or is it all undergraduate?
We not when I first came.
Okay.
We had an interdisciplinary graduate program, but not a biology one.
Okay.
I think I was here 5 or 6 years before we had one. And so I used undergrads, and they did a lot of the work. But what happened, because it was a small place, when they finally did they got a new president in, and he was interested in encouraging research. So he started a small in-house granting program, and I was able to get funding from him to go do projects. And so I ended up using those funds, plus I could get some grants from zoos or different places. And I actually set up a small endowment too. But what I did was pick projects that might, they produce a paper, but I get to go do some fun stuff. So I actually started doing that. And it wasn't deliberate. It just kind of happened. And like the first one was going to Haiti. I met Bob at a scientific meeting, and he told me about these 3 species of Euromacer. Two of them have pointed noses, and one of those is brown and one is green. And then one of them has a round nose, and it's green. And the round nosed one chases frogs and eats frogs, and the other 2 eat lizards. But they stalk them. And he said you never find the brown pointy-nosed one and the green pointy-nosed one in the same place. So at any rate, he invited me to go along and bring some back to look at what they would eat and how they chase them. I got into the animal behavior aspects of it. So I ended up getting to go to Haiti and spent 3 weeks down there with him and collected these Euromacer and brought back and did a project looking at their stalking behavior, how they catch their food. And I'm trying to think where Rich and I went to Arizona on one of the trips, and I went up to Manitoba to see the dens up there, and got most of these I got a paper out of, but it was oftentimes just one, or it would be something. I guess I didn't have the track record that it would take, of doing the same thing like pheromones over time, to where you could get an NSF grant or something like that. Mm-hmm. But what I got to do that was, I think, more fun was I got to do lots of different things. I might study Euromacer, stalking behavior and garter snake pheromone trails and cottonmouth field projects, and I got to have a great 40 years of doing that. And whereas if I'd have stayed just doing maybe the phenotypic plasticity for my whole career, I could have probably succeeded in a different way, getting grants and so on. But instead, I got to go to a lot of places, and I actually during COVID, I ended up going back and getting all my field numbers. Because one of the things that happened when I was at Kansas, I took a course in museum techniques along with John Simmons and a couple others, and we learned how to do things for a museum. And one of the things that the professor taught us was keeping a journal and how you do it in science and all of that. Well, so we had to do one in that class, and I've done one ever since. And so, I went to Southern California. I'd sit on a rock and look at lizards for my project, and had plenty of time to write in a journal. Well, during COVID, I had all of these journals that I'd been wanting to look at again and maybe summarize or something like that. And so I did. I started pulling them out because I was stuck here in front of the computer, and ended up summarizing them and putting them in my Facebook account. And people would write back and say how much they enjoyed them and all this kind of thing. And I don't know how it came about, but somebody probably suggested that I should turn them into a book. And so I did. And I put in pictures. I'd taken slides on all these trips too. So I put a lot of pictures in this book. And the university was, again, somewhat fortuitous, looking for books to publish. They had a few in some of the education areas, but they didn't have a nice collection of authors' books that they could sell. So the University of Texas at Tyler published this, and I got to work with them very closely, got to do them exactly the way I wanted, and wrote the book where I talked about the field trip and how much fun it was and what we did and all that, but also about a lesson that I learned during that particular trip. Each chapter had something in it about, learning how to collaborate with your colleagues, or different things that I'd learned on those trips. So that was very fortuitous again, but one of the most enjoyable things I've done in a long time.
Yeah, so it's basically a book that kind of, catalogs your career and the places you went and the things you've seen, and
from a travel point of view, yes.
Yeah, and so what's the name of that book?
Adventures of a Field Biologist.
Great. And where can people, if people wanted to read that or buy it somewhere, where could they find it?
Well, you can get it from the university, but it's easier just to do Amazon.
Okay, great. Well, we'll put a link to that in the show notes, so everybody.
And I don't make any royalties on it, so it's they can. I'm glad for them to read it. Everybody's enjoyed it, but I don't actually get any money out of it. Well, I was more interested in getting it out, mm-hmm. and then trying to find a publisher who would give me a good deal on it.
Yeah. Well, so I'm assuming it supports the university to some degree. So yeah, again, I'd encourage everybody to read it. Well, before we wrap up, there's a lot of things we could keep talking about, but I would like to talk about there's a gathering that happens every so often called the Snake Ecology Group meetings, and I know you've been involved with that, pretty much right from the beginning, and I've been involved in it somewhat, but I'm curious if you could kind of talk about those Snake Ecology Group meetings and how they came about and kind of the role you played and those types of things.
Well, it was somewhat serendipitous too. Pat Gregory was the host for a American Society of Ecologists and Herpetologists in '89, I think it was, and I offered to do a symposium at that meeting with Pat, and so Pat and I organized a symposium on snake ecology and behavior, and put it on at that ASIH meeting in Victoria, University of Victoria in Canada, in British Columbia. And we had so many people come to give talks on snakes that Pat had to open up a whole nother day to accommodate them. And so after the regular meeting, we held a kind of a off-the-cuff get together of all these folks who were interested in snakes and ecology and behavior. and talking about what's the future like, why are they working with snakes, and we said, well, let's see if we can keep this going, and have maybe some regular meetings or something. Well, Chuck Peterson and Ralph Gibson volunteered to put together a newsletter with all of our addresses and all the maybe little things on what we were doing research-wise and so on. So they put out a newsletter and mailed it out in those days. And we had two of those come through, where the second time around they got more people, and so it gave addresses of a lot of snake ecology people. So nothing happened for a few years, but then Rich Siegel volunteered to put together a group and have a meeting in their field site for southeastern Louisiana, and that was on an island in Lake Pontchartrain, and they had a house there, and so he took those lists and put together a group. He could only handle, I think it was about 50 people. He just didn't have space for more. So we had our first one there at this field station, and it worked out really well because nobody could go anywhere. You had the food brought in, you had to leave by boat if you were going to go. And so everybody was stuck, and we ended up spending a lot of time brainstorming, and everybody really enjoyed that. And one of the things that came out of it, Rich wrote he and a couple others put together a book on snake ecology and behavior. And everybody said, we ought to have regular meetings of this, because we can, and do it at field sites where people are going to stay and talk and brainstorm. Well, nothing happened for about 6 or 7 years, and then I put one on here in Tyler at a school camp where there were bunk beds for 120 some people, and we had that many people, and the internet was email was just starting, to where I could send out messages about this meeting. And when I started, nobody, they said, well, I can't come. I wasn't getting much response, at least not many of the people that I really wanted, the ones that were doing good work in snake ecology. But what happened was I got a couple. Rick Shine, who said he'd come, a couple others said they would come, and by having email, I was able to send out periodic messages to everybody about who was coming. And once a few of the big names started to say they were going to come, everybody else changed their mind and started, oh, okay, I'll come. I'd like to give a talk. And so it ended up being a really fantastic meeting. I had people from Israel and Sweden and Japan, all over the place. And it ended up being great. And I think, in my view, the best of all the ones that we've had. But again, it was somewhat fortuitous that it just happened to be that email came out about them. Well, what's happened is we didn't really become organized in such a way that there were any regular meetings. So what happens is we've had several folks who just wanted to do it. And it would be between 5 and 7 years in between. And just they ended up working out really well. They were always at field stations where people would end up, spending time with each other and collaborating on ideas, and ended up being a really fun meeting, very different than any of the others. And a lot of folks like that. There's typically only 100, 150 people instead of the thousands that you get at some of the bigger meetings. So So we had our 7th one this year, and it was up in Winter, Wisconsin, which didn't seem like a very logical spot, but it ended up working out really well. And we had a lot of folks show up.
Good. Excellent. Great. Well, I've enjoyed the ones I've been to. So, it's a great thing. And I encourage anybody who's works in snake ecology to think about attending the next one or maybe hosting one in the future. So anyways, well, we've been going about an hour here, so I want to begin to take this one in for a landing. But before I let you go, I'd like to have all of my guests tell me a snake story. So Neil, what's your best snake story?
Well, probably they revolve around some research project. And I guess one that I like to tell the most is a project that we did with pheromones again. But I was at a meeting and I was presenting material about snake trailing and that kind of thing. And one of the things I mentioned at the end was I didn't know how the snakes knew which direction to go, the male snakes, when they were tracking a female. How do they know that they're not going backwards on her trail when they encounter it and follow it? And I just didn't have a clue. And so I mentioned that at the end of a meeting. You know, oftentimes you have an opportunity for questions. Well, I asked them a question and said, hey, does anybody have an idea as to how the male snake knows which way to go? And so I don't remember if it was right then or somebody came and talked to me later. But somebody came by with an idea and said, well, think about how snakes move. They have to push against objects in the environment. And so when a female is moving and she encounters a rock, she's going to go around, she's going to push on the front side of that rock. And then the next one down the way, she'll maybe go on the other side, but she'll push on the front of it. And one of the things they didn't know this, but I'd seen male snakes would come up I used pegs in my apparatus. They would come up to the pegs and they would tongue flick both sides of the peg before they would move on. They would tongue flick the peg a lot. And so this guy suggested that maybe they're using that orientation of having more pheromone on the front side of the rock and not on the back to get the direction. Well, it was real easy to test, and I put an undergrad on it. We had an apparatus and had pegs in it. And we put straws over the pegs because we didn't want to have to wash them all the time. So we had these pegs that were just the right size for straws. And so we cut pieces of straws. Well, what I did was I taped the student did, taped the straws down to the pegs, ran the female down we ran a little track. And so she touched, made a track on the ground, but also touched the pegs. And so when I released the male, they would come up and tongue flick the pegs and go one way or the other. Well, the experiment involved taking the straw and turning it around 180 degrees. And so I had some of them that were tested with the tape on them, and they would just be normal and they would go the right direction. But if I turned the straw around 180 degrees, they went the wrong way.
Wow.
So
that's really cool.
This is an undergrad. He gets a paper out of it, ends up going to medical school. But yeah, it was just one of those that I and the sad thing was I published it in the Journal of Chemical Ecology, which had just come out. It was a neat journal, but it had no readership. And so I published this in there, and it made drawings of how it worked and everything. It was so elegant a paper, but nobody read it to speak of until Bob Mason wrote a review article on pheromones in one of the biology of the reptilians, and he reproduced the figures that I used in my paper. And then all of a sudden, people started to read it, and I got a lot more attention at that point. But it didn't really matter. It was just such an elegant little project.
Yeah,
and so there's my story.
Yeah, it's great that you got that info, kind of at a conference too, kind of throwing this question out to a group of people. And I love that. It's great. So,
again, serendipity, by the way. Yes, so common in my life, but also I think in a lot of science.
Yeah, exactly. Great. Well, I've really enjoyed learning more about your career, and I encourage everybody again to go out and read your book. And but yeah, just thank you so much for joining us on the podcast today.
You're welcome.
And I just wanted to thank the audience and tell everybody to remember, snakes are animals too. And it's a privilege to see one in the wild.